Okay. Azim, what do you think we should do today?
So today, I think we should discuss about the sequencing. So why do we do sequencing?
Okay.
What is the need to do sequencing?
And what is sequence?
What
What
is the
is the
sequence?
sequence itself,
What is
right?
the sequence itself?
Yeah. So I'm going to have to, this is a very basic question. I think this is good that Azim has been trying to do this for some time, and we are going to make this first video addressing the very basics of bioinformatics. Okay. So thank you, Azim, for bringing that question. So for this, I have to take you guys back to 1953 when the DNA structure was solved. All of you would know. Watson and Crick solved the structure. The structure was not just a structure, it really created molecular biology as a discipline, because that's the first time there was any idea that DNA could be the genetic material that is responsible for life. Okay. So, because of that, there was a lot of interest in that. And slowly, people were thinking maybe proteins are the functional thing. That was known for a long time. Biochemists have been active understanding proteins, because proteins are very visible molecule, they're jumping around doing something, so we could see it. Okay. So DNA was sitting inside the nucleus, nobody could see it. So people thought it could be protein. That is the genetic material. Then when the DNA structure is solved, first time we know that DNA is genetic material, then the question comes out. We knew amino acid, we knew proteins are made of amino acid. Then we're looking at DNA, that's made of nucleotide. How does it happen? How does the nucleotide become proteins before it functions? Right? That's what I'm talking about. Do you want to add something here?
Actually, the entire process when we speak about mRNAs, which bridge DNA to protein, and the axis is tRNA, ribosomal RNAs, and we only have mRNA playing the part which is getting transcribed.
Yeah.
The switch.
So that's what it is. So now this takes me to central dogma of molecular biology. How does the DNA become protein? Is DNA even coding for protein? If it does, these are two different languages like Hindi and English. So someone has to translate. Okay, we knew neither the languages. We didn't know the language of DNA. We didn't know the language of protein. So we're looking for a Rosetta Stone, really. Okay, to translate from one to the other. So entire 60s, I would say, late 50s and 60s, we were just trying to put this puzzle together. And one of the biggest thing was to, even before the codons came, we knew that central dogma of molecular biology, DNA, becomes as Drupad was mentioning, it transcribes into RNA, and that translates into, we quite didn't know how, but that dogma was put together because DNA is inside the nucleus. Proteins are always outside, functioning, roaming around and having fun, right? So this nucleus, how does it, and the DNAs are huge. DNA is a single molecule, longest molecule chemistry, longest chemistry. That's there. You guys would know, like in human genome, we have chromosome 1, that's hundreds of nucleotide long. Okay, that's the longest molecule. These are not A, T, G, C. The A is connected to T through a sugar group. So it's a big molecule. So you have to keep that in mind. When you're saying sequence, you have to just know A, T, G, C. Don't get into the alphabets. It's all chemistry, right? So then it's such a big molecule. How does it make protein? Proteins are small units, right? So that's an issue. That's where the in between the transcription came. Oh, it must be coming, part of it is getting transcribed and then comes out of the cell and then get translated. All those nitty-gritty detail, I don't know how people did that and addressed that so patiently. They're all there in those papers. But once they did that, then still DNA to RNA, it was easy. There's only T to U. So, somehow that's getting managed, right? But RNA to protein, that's a totally different language. That's where Corana and Nirenberg and everybody came and said that 3 nucleotide, 1 amino acid. Okay, that's when the codon table came up. Okay, all of you, I know during interview we ask it all the time, like, what is degeneracy? You're right. What is degeneracy, right? So that's the biggest thing you want to talk about it?
Yeah, so
Yeah.
degeneracy is basically, so there are a lot of codons. So multiple codons can just code for a single amino acid. So that it's redundant for us as when it comes to the structural prospect. So so talking about the central dogma, so the DNA gets converted to mRNA, which gives the ORF for it to be translated to a protein. So we get two different type of sequence. One is the nucleotide sequence and the other is a protein sequence. So the nucleotide sequence will consist of ATGC and the protein sequence consists of amino acids. So now, is there a fundamental difference between these two types of sequences where it is more relevant? Like,
Yeah.
so ma'am, if you
So now just going ahead with that central dogma, degeneracy. I was just talking to them during lunch. If you had you can imagine a replication system that's perfect. Okay, no error. 1,000 times it does exactly. Perfect machine. Nature didn't make that protein system. Why? Because there will be no life forms. There will be no expansion of protein function. There will be no blast. There is no need for blast. We will not even be sitting and talking, because we won't exist. Correct? So replication, errors happen in the replication. That's where the degeneracy comes in. When replication happens and there is an error that went into the triplet, we told you the codon is triplet. Each triplet represents into one of the triplet, like which was supposed to code for alanine, let's say, one of the codon got error while replicating. Then that either the amino acid changes or it is in such a position in the triplet that the amino acid doesn't change. Here I'm going to go a little bit detail. The codons, if you look at the codon table, there's a first position, second position, third position. The third position is called the wobble position. I was so impressed. I want you guys to go look at it when you go to the class. Most of the accepted mutation by nature are in the wobble position because the other positions make it change the amino acid. Okay, so serine, for example, all four nucleotides at the fourth position will keep coding for serine. Similarly for leucine, similarly for arginine. So for third position, wobble position, nature just selected. I'll ignore it. Do whatever you want. I'll manage somehow. Right? So that's an interesting part of it. Every time I try a mutation, that in a patient or something, that will be in the wobble position. Okay, even if it is a unique position. Sometimes it is deleterious mutation, like aspartic acid changing into lysine, which is a very different positive to negative charge and negative to positive. Those kind of changes, they're deleterious. Those changes are not in the wobble. It will be right in the middle of the first.
And the frame shifts as well.
Yeah. So sometimes it deletes and frame shifts. So then whole protein get destroyed. One base goes. Why would the protein get destroyed? Tell me. Because it's 1, 2, 3, 4, 5, 6, 7, 8, 9. That's how it's going. Suppose 4 got deleted. Okay. Then 1, 2, 3, 5, 6, 7. That's a different amino acid. That could be a stop codon and it's dead. Protein is dead, and the nature didn't select it. Okay, nature selected those who did not delete. So that's frame shift. We'll get into those things a little later. But going back to degeneracy, nature kept on selecting tolerable mutations, and because of that, there's function expansion. Some it tolerated and say, oh, okay, this tolerating means what is that tolerate? It is tolerating the 3D structure of the protein. If the 3D structure of the protein did not tolerate a mutation, go ahead.
The structure of the protein determines its function. So if the residue is not conserved in that structure, then the protein's function won't be there.
The domain will change.
Yeah, so the function itself will change. So everything is for the function. The structural aspect, the sequence arrangement, everything is for a particular function.
Yeah. It's a natural selection when we're talking about it selecting the function.
Functional.
It doesn't even care what happens to the protein. As long as, even if it's not a protein, something else I put there, spaghetti or something, as long as it's functioning like an immune process, it'll take it. Okay. But when he was talking about function preservation, And protein structures, it may be a little soon to say about it, but proteins are folded like rock, right? So some amino acid are inside and some amino acid are outside. So when I'm talking about nature tolerated, it tolerates more changes outside of the protein because it's not required to make a solid structure inside. You will learn about that. So there is core amino acid. So core amino acids are more likely to be not tolerated, any change. It will be very fussy. On the top, and the top is the one for function. The top is the one that's required for function. So sometimes because of that, it can take new functions. That's when function expansion happens, right? Okay. So
So another question I have, ma'am, with regards to just the sequence. So
Yeah,
a change in amino acid versus a change in the nucleotide. So the impact would be different depending on the position.
yeah.
So so depending on the combination of the codons that we have, so how many different types of combination for a particular length of protein we could have?
Okay, so there you're saying that
Yeah, so
amino
say if
acid-
I give you a protein with say 10 amino acids, now how many combination
DNA, okay.
of
So we were talking about it this morning, right? Like
yeah,
it's innumerable. That's also another point
so
we're missing here. Like
Reverse
for
translation.
example, for example, you have a DNA. You have a DNA. It will only translate into one protein. It could translate into three proteins because I have three frames, and that will come later. But it will always do only same protein, same triplet, same protein. But the question is, can you go from protein to say that which DNA is it coding from? Here is a whole gap. Okay, because that's very important even to, if you're dealing with sequences, you need to know this difference. So I give you, you can write down, I'm not going to give you now, you can write down a 10 amino acid stretch. You know, there are 20 amino acids. You write down in a notebook as you're listening to that, you can write it down. And then what can you do? You go to the codon table. Okay, and for each amino acid, you can say how many codons are there. Then all that will have coded for the same amino acid, then keep multiplying it. Okay, if you took 10 amino acid, 10 numbers you're going to multiply. That will go into tens of thousands. Okay, so tens of thousands of unique DNA are going to produce the same protein. Okay, so that gives you now, that's where, although DNA, proteins come from DNA, that's a point I think Azim was making, like DNA kind of goes this way and
so
protein
there
goes
is a difference between a mutation at a protein level versus a mutation at a DNA level.
DNA level.
That is very different.
DNA can take, large number of proteins and tolerate and make exactly the same protein again and again and again. Okay. So, in fact, some proteins cannot tolerate, I don't know, because of fold of function, they don't tolerate any amino acid change. They're like 90% always similar to among even different organisms. But some protein, immunological molecule.
Yeah, maybe that's because of the functional aspect of the protein.
Yeah, so the selection is happening at the function level. If function changes, it's not going to like it. Like I think polymerases, for example, they're not that divergent at all. They're very
You have to talk to
So they have the function that very primary. So sometimes with very divergent protein families, you can tell from the diversity that they are probably not so much required for the key functions. Like key function, they all conserve because they cannot afford to change it. So in any event, that's going too far from here. But you got that point, Azim was making is DNA level mutation has a different story on its own. Protein level mutation is not that much, and that has a story on its own. DNA level mutation, I'll tell you. Because DNA diverges so far, okay, sometimes they probably don't recombine and speciation happens. But the proteins is still the same. So that species also have the same proteins like mouse. We all have ribosome, we all have, digestive system and everything. Because that's how DNA, that's another way to look at DNA
Yeah,
diversity.
exactly.
You know,
It's like different signature, signature at the sequence level, but at the protein level, they are way similar than any other.
Yeah. As long as it DNA diverges enough that they cannot recombine, and then it will make another speciation will happen, right? That's so replication error has been great because we have so many beautiful species, millions of them in the world, and we are complex. We are complex. That's also another thing I will tell you from the same point as mutation at the protein level, DNA level, suppose it's going to mutate, how does the function expansion happen, right? So what nature does is it copies a gene duplication, genome duplication. And when the duplicate gene is existing, then it can do experiment with the duplicate gene. This gene is doing what it needs to do. So the organism not killed. But in the duplicate half, it can play around with many more changes until it picks a new function. So that's called paralogs, right? That's how we got so complicated. We're so complexity is so high, right? So mutation is a lot of beautiful thing. Now that's why we have so many beautiful computational method to understand it, because we have no clue how to handle this. Nature has done it so beautifully. This is where I'm going to tell that part,
Yes.
where I'm going to say that what you can do in computers so easy, nature does it complicated. I tell you, so if I write a DNA sequence, A, T, G, C, 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 A, G, whatever, right? And then you are a programmer, you know how to write simple programs. So you're going to say that I have a codon table. I have the codon table, I have the DNA sequence. If the DNA open reading frame, I can mention it here. Right now, I don't think it will be too complicated, right? Is it?
Okay.
So if there is a stop codon, that's not a protein, right? So open reading frames are when things keep on going for larger, to make large enough protein, 100 amino acid, that kind of thing. So you know the open reading frame. You can come up with a protein sequence. So if you know the DNA, you can come up with a protein sequence, but with a little bit of AI or whatever you call it, just regular intelligence, right? You can say that, okay, this is a protein that's making. So that's so easy in the computer. But nature has to do so many steps. It has to make mRNA, it has to make tRNA, it has to make tRNA synthetases, RNAs to come together to form ribosome. So many complexities. And then 2 amino acids will barely come and stitch itself. And then 100 amino acids come to make protein. So complicated. In computer, forget it, ma'am. I'm going to just translate it, right? Give me the DNA and you go away, right? You get the protein. That's good. So, but now this is where our problem starts, where nature can do it so easily. Okay. So did we cover everything before this, or I'm jumping too far?
Yeah, almost we have covered
Almost there?
everything.
Okay.
So
If
we have to,
missing
yeah.
something,
So
just
just
let
I
me know.
have a question.
Yeah.
I want the viewers to address it. So why do mutations happen at a DNA level? Is it due for functional change or stability or any other aspect? So you guys can answer that. So now we can move.
You can, yeah.
Yeah.
So you guys can answer that, discuss it with your mind and with your peers,
so-
next to you. You think about it. Okay, why does mutation even happen at the DNA level? That's it.
Yeah.
Yeah.
So
So-
since it's a blueprint, so every
yeah.
changes has to be there.
Yeah.
So why do that change happen?
In the first
Yeah.
place. Okay.
So-
So that you guys can think about it. You got enough AI based tools to dig in, right? So now I'm going to say, where did I leave? I left at the point where I said that it's so easy to go from DNA to protein on the computer. I just write a simple code, and that code will work the rest of your life, right? I can borrow it from Azim, can borrow it from you, and all that. And the nature does it so many steps and so many proteins it needed, so many catalysts it needs to make 2 amino acids come together. Now, what nature does it so easy, once it makes a protein sequence, which is protein sequence is actually a chemistry, right? It's a macromolecule. It's connected, and it will fold immediately like this. It will fold into its perfect 3-dimensional structure. Every time you open it, it will fold exactly like that. That not will happen in your dinner plate, right? If you put spaghetti this way and then you change it and then come back, you can never get the same configuration for spaghetti. It will be different. That's not what happens. It's not arbitrary. The information for getting into that fold is there in the sequence itself. That has been known for some time now, but we don't know how.
That is a
We
problem.
still don't know how.
We know the end state, just
Yeah.
how
Yeah.
the protein looks.
That is the bigger problem in biology that needs to be addressed.
That need to be addressed. Maybe that's another next Nobel Prize. We can assure that it's whoever solved it. We don't know how it does it. within seconds it will fold into its native structure, and every time it's folded, it looks like complicated for us. But how does it know it has to exactly, each atom knows where it has to go? That's amazing. Okay, and that how it folds, and we know the final structure as a thing was saying, that comes from it's come from an experiment called crystallography. We'll come back to that. So, but now nature does it so easily. Okay, but we still don't know how to fold. You give me an amino acid sequence and 1,000 computer, all the servers in the world. I cannot fold it. So what nature does so with so many steps, we do it so easily. What nature does so easily, and that is where our whole bioinformatics sitting. Classical bioinformatics and structural bioinformatics is an area where a protein sequence is there, how it folds, and what function it's going to assume. That's the biggest question. You want to add something, one of you?
Yes, ma'am. At least, I mean, when we put the sequences together, we get to know that okay, this part looks like it is conserved. Maybe they are performing similar type of functions, and domains forming.
Yeah.
Doing the
So it's
categorization.
mostly a pattern recognition that we are
Right.
doing.
Yeah.
So we will come. So pattern, if we are seeing a pattern that is conserved in a sequence, that means there has to be a functional aspect of it that is preserved with those conserved sequence. So now we'll proceed with the functional aspect of a protein's sequence. So
Since we cannot
Yeah.
predict from the sequence itself, no matter what.
Yeah,
Even with the signature, Madhurpad was mentioning about signature, and you hammered on that. Even with the signature, if that signature, I'm seeing it for the first time, then of course it's not a signature. I don't know what it does. So what we're still doing is by comparison. If I already know a kinase, somebody did the work before, I give them 1,000 thanks and I tell them, thank you very much. Mine looks like a kinase because I'm sharing the same signature. That is really the basis of sequence comparison method, right? So from one known to the other known.
So that is where we use BLAST.
That's where we use BLAST. BLAST is also in a way is a local sequence alignment.
Yeah.
So that tells you that I found the signature. Oh my God, I know it.
Yeah, so
Okay.
we just check the similarity between two sequences using BLAST.
Yeah.
So
in our in the previous BLAST podcast when we were doing, Pushpinder and I talk about this. Pushpinder asked me a question. I said, so if I get a hit in a kinase in BLAST, can I tell it's a kinase? Okay, I'm going to repeat that answer again here because it's relevant. Then kinase is not signature. That signature tells you it could be a kinase. Kinase is a huge molecule. You can't tell from the tail of the snake that it is a functioning snake and run away from it. You have to the whole function has to be there. Whole snake has to be there. So then you have to see that okay, it has all the domains and everything. So it takes more work after BLAST.
Yeah, we have to look at more of a functional side.
Yeah.
Just taking the whole.
Yeah.
For the function aspect.
So the signature is signature, maybe, but we were talking about it folding. For folding, you need signature and some other thing coming with the signature to fold, right? So that is a full length protein. So we want to make sure that there is no stop codon in the full length protein. Everything is translated. It's open completely. Then you say, oh, looks like it can be kinase. Of course, then you have to. The final thing comes in the experiment all the time. Okay. Don't fool yourself, finally, because I'll tell you one example of what happened after. Every time I have to tell a story, okay? So maybe these guys didn't even hear it. So one of the stories is when human genome was sequenced, completed in 2001, okay, before that, kinases are important target for cancer. You're working at the cancer thing. So cancer targets, okay, very big cancer.
For every pathway, kinases are
Kinases are and many kinase inhibitors are now in treatment, like Gleevec and other things for EGF receptor and so whole world, especially Such's company, they were looking at for kinases. They wanted to know all the kinases in human. So there is a paper in 2002 that calls Human Kinome. Okay, when they published that paper, they got it from simple snippet using BLAST. Okay. And these are not even known. They're not known kinases. You only saw it. It could be pseudo gene, they call it sometime. It may never form a gene, but then they had to find all the fragments. Then they had to clone it. They cloned all the 518 kinases before publishing that. They not only had the sequences in there, they had the clones of all of them, so someone could add it. So that's a final verdict. Final verdict. So bioinformatics never
Functional
stopped.
validation is required for everything. So,
Yeah. Functional, it even folds.
Yeah.
You know, even before it has to express in the fold, it's coming out. Okay, that's of course function means it's folded. That's another thing, right? So that's how far you have to go. But you guys don't have to worry about it at the time. But if you found a protein that you're really, really interested, you need to either you yourself have to do wet lab, or you have to convince somebody to follow it in the wet lab to get to the bottom of it.
Yeah, but ma'am, doing one by one blast would have been really difficult.
Yeah.
Yeah. So
Yeah.
is there any other method that can be
No,
used?
the kinases I was telling you, they will use kinase HMM. Okay, so the model is there that's made for HMM. Then they will use that model to search the database. All the kinases will come. Every morning you have to do, because every morning there are new sequences coming.
Ma'am,
Because
but if we could tell that there is a pattern of, say, alpha helices and beta sheets, a particular pattern which is conserved across multiple protein
that
sequences.
secondary structure, because alpha helix is there in so many proteins.
I mean, a particular pattern of, a set of alpha helix,
Okay,
beta sheet,
yeah.
alpha helix,
For
alpha
that you
helix,
need to
like
have full length, no?
right.
Yeah,
That-
full length, that's a different thing.
But
If you have full length protein,
but
then
easy method would be just aligning all
Multiple
sequence,
sequences.
multiple sequence, and just telling that this region is conserved.
Yeah.
That is the easy method.
Yeah. So first, for finding the blast signature is the best way to discover. Okay.
Yeah.
In gene discovery, that's the best thing you can do. And then you're putting full length.
Mm.
When you put full length, you already know it's a kinase. Then you know how the secondary structure
Structure.
pattern is going to be. So that, but what Dhruv is saying, Drupad is saying is a good thing. You have the complete protein. You know nothing about it. You're not getting any blast sheet. You're not getting anything. Okay. And we can't even predict the protein structure. But what we could do, as you were saying, alpha helix and beta, and somehow try to get a feel for what it could be. Okay. And is there a protein that's known where this pattern exactly exists and somehow related? Those are all difficult things, but it's doable. But depends on what you have in your hand, right? And so, having done that now, okay, we're talking about fold and function, fold. Why does sequence alignment comes in here? That's a multi-million-dollar question here. So, because fold is what keeps the function, then why don't we just compare the folds? Simple, right? And who gives you fold? Sequencing machine churns out the DNA. Sequencing machine, so your computer can churn out the protein because you have the DNA sequence. You're really smart at that now. So you have, say, billions of proteins, you churned it out. Okay. But we don't know the folds. We don't know all the folds yet, because structure prediction and structure experimentally determining structure is still a very arduous process. Okay. And so, for example, if I have a protein, first I have to have the whole protein. If you have DNA, I cannot do protein structure. If I have the protein in the computer, I cannot do protein structure. I need actual protein. Someone must have really expressed it. Recombinant, right? You guys must know about recombinant technology. I'll tell you a little bit about that, because that's the one that really stimulated protein structure and other things. In recombinant technology, which gave birth to biotechnology, what did they do? Here in the computer, you're going from DNA to protein, right? Easily. They were saying, okay, you give me the DNA. Okay, I will put the DNA in a construct and put it in the yeast or even E. coli. There's some organism yeast. You put it there and just yeast grow, just like you're going to grow bread or idli ka dough. Just let the yeast grow. Or you're making beer, let it grow. Every time it grows, it keeps making that protein. And you can purify it using antibody to that or some other tag. You can get the protein out. That is recombinant technology. That is what gave birth to the whole biotechnology industry. Molecular biology is one thing, biotechnology came because I was there when that happened. Genentech, when it started, that's the first recombinant technology company biotechnology, and Paul Berg was probably working there. I'm not 100% sure. He just turned 100 years this year. Okay, 100 years of recombinant, 100-year-old now. So we celebrated recombinant technology. So if that's something we can do. So why I'm talking about recombinant, certainly biotech and all that, because you need large quantities of proteins to experimentally determine the structure of the protein. You get large amount of pure protein, and then you have to crystallize it, and crystals have to form, and single crystals have to form. People spend their lives just looking at the crystals. Oh my God, I didn't get a single crystal. This thing is ruined. They will have actually trays of, Tyagarajan's lab, they will have the whole tray, right? Several pH conditions, several salt conditions, several, ion condition. They will be doing it simultaneously. One of them, they will see a crystal. Then that's your Eurocade, okay?
But when we do have the sequence of of say two proteins, can we say that they are maybe going to say they are 70% similar, they are showing identity. So can they fold in a same way? Like, can we say
that's
that?
that's what from sequence itself we are able to say because of the signature thing. But then we'll go to that because that's where AlphaFold and everything is going to be helpful.
We will go to that, but
we'll go to that.
We'll just come back to the sequence part.
So now you have now you can, if I had the crystal ball, that's something I always say. You give me a protein sequence, I have the crystal ball. I'm going to close my eyes and say, I got the 3D structure. It's a kinase. I can't do that, unfortunately. Neither the experimental techniques are fast enough to do that. So can you say what is the closest we can do? Because if I know two things, as you were saying, two things fold the same way, I know they're functioning the same way. Okay, at least I can. That's the best because closer to function is a fold. Okay, sequence, folding, and then function. So closer to function is a fold. So fold is the best way to do it, right? So we can do that because there is a lag. Okay. Then we're saying, as Azim was saying, the signature you're talking about, small signature. During the mutation process and accepting it, some core in the core of the protein, it is less tolerant of mutation. Because it has to fold to function, I told you, right? So on the surface, it may be more tolerant. Natural selection is more tolerant on the surface. But in the core, it's not tolerant. So sometimes, even though two protein may be slightly doing different function, okay, they are paralogs. One is a JAK kinase, another is something else kinase, but their core is similar. So the remnant of that signature, because natural selection left that remnant, because if that gets destroyed, the protein gets destroyed.
Ma'am, also can we say that it is because of the core which remains conserved, it acts like a seed or a nucleation center on
Definitely.
which
It's a fossil record, you can say. It's a fossil record of where it started, right? So it's a fossil record, and that's why you have profile-based search, this and blast. Any sequence-based search depends on that fossil record that is left. But one thing I will tell you here is that fossil record sometimes even loses the fossil record and they still fold the same way and still share the function. That's a limitation of all sequence-based methods. But we can go really deep with blast and other things. And then after blast, okay, we did the blast lecture, and I'm going to assume now that you know how to use blast. When you get a small snippet, you evaluate and everything you check, and if you're convinced, then you look at the full length. You got to keep on going, okay? You can't stop right there. Oh, I found. Don't talk before you really found it, okay? So you be careful about that. Then after that, what was I going to say? We were going to jump into
multiple sequence.
multiple sequence alignment. So now, I suppose I have 5 proteins that I already know based on some other work Azim has done and Drupadat, and I know that they all belong to the same pair. They all went through hard work, blast karke, full length leke, sab karke, we have this. We have those sequences. Those 5 sequences now have some similarity in the core region. That's how we found it. And they're not similar. So you can do multiple sequence alignment to see how it got preserved, what got preserved and what didn't get preserved, what nature tolerated and what didn't nature tolerated in that particular fold. So you're not even looking at the function at this point. You're saying that all of them are same fold. Nature has somehow smartly selected them because they selected for function. Now you're saying by multiple sequence alignment, you're saying that, okay, which region the nature could not tolerate. Okay, so you get more information about the fold family rather than the functional family.
Yeah, it's rather than just doing two sequences at a time, we'll do multiple sequences
Yeah.
just to see the similarity between the functional part.
And ma'am, if there are gaps,
Gaps will come to the gaps.
Yeah,
Like
so-
that's what multiple sequence alignment. When I'm saying, Drupad, this multiple sequence alignment, I was telling you about the core and I was
Core.
telling you about
the outside,
and the
Yeah.
loops and all that.
Mm.
So in the core region, you won't have gaps.
Yeah, so what needed to be preserved is always preserved.
Preserved. If a
Yeah.
gap is there, then one amino acid is gone, then the whole thing shifts. Then it won't form the core anymore.
Yeah.
For example, I'll tell you in beta sheet, I don't know structure work, we will come to that, those kind of, but in beta secondary structure, one is pointing in, one is pointing out. The one that is pointing out is hydrophobic.
Yeah.
Okay, one that's pointing out is hydrophilic. Suppose he deleted the next hydrophobic, then it's a peptide bond. Okay, so it has to come and turn around and put the hydrophilic inside. Oops, stood up. Nature wouldn't accept it. Okay, so that's why you would have more conservation, like thick block without gaps.
In the
Okay,
nuclear
without gaps. That's what multiple sequence alignment is the first time, for example, Drupad, thanks for bringing it. First time it tells you where nature accepted not only which amino acid, which regions it accept more tolerant, which doesn't is not. The more tolerant regions have more gaps. And there'll be length will be different. This will have only 3 amino acid before it gets into core. One of them will have 7 amino acid, goes all over the world and comes back to this. So there's a lot of difficulty in the loops. That's why they also take different functions because loops are functional.
The
Right?
loops determine like
mostly the
whatever
active
yeah,
sites.
so variation is because of the loop.
Yeah.
Loops.
So multiple sequences, as Azim was mentioning, there's a method called dynamic programming algorithm, okay, which is a very smart method. We're not going to go into details of that. We're going to assume it is doing a wonderful job, even with gaps. Okay. 2 sequences, but 2 sequences of the same size. This is the mistake people make. If you give one sequence only 5 amino acid and the other sequence has 100 amino acid, dynamic programming will screw up. It will do the 5 amino acid, it will rubber band. It will just rubber band the whole thing with gaps. So dynamic programming method does a good job, but you have to give it 2 proteins that you already know that are similar. And the folding domain. That's the input to dynamic programming algorithm. It's not God.
But if we are doing, say, local alignment, and we go and we try to say, if we know that this domain is related to this function, and then that particular domain we are trying to map in the genome or like in the protein, the full.
That you can do
that
it.
is-
Dynamic programming algorithm, there is something like local alignment. Smith-Waterman does a little bit local. I'm not going there because we're going to go into multiple sequence alignment. Okay, I'm going to leave that detail.
Someone wanted to ask a question about
Yeah.
that. So, since there are 2 types of sequence, one is nucleotide sequence and the other is protein sequence. So, in what conditions will we prefer using a protein for multiple sequence alignment, and in what condition will we prefer nucleotide for multiple sequence alignment?
I don't know. I have only one application I can tell you where multiple sequence alignment of DNA is even useful. Okay, correct me if I'm wrong. That I was giving an example yesterday in the class. So you have a signature, protein signature. Okay, you found it like 5 different proteins of the same signature. And you want to make a PCR primer of that. Okay, now because you cannot reverse translate the sequences into DNA, what you're going to do is to take the DNA corresponding to this that's coming from human, take the DNA corresponding, and then juxtapose them, not even align them, juxtapose them. You will see that although 1,000 different other ones could have coded for it, you will see that the G is always conserved because nature never allowed the G to change into C. Okay, so there is some conservation within that. Although the protein will look really weird, but when you look at all the DNAs, then you can make a degenerate
I
primer.
think, ma'am, there are other regions also because
Like the
there will
Tata
be
box.
so the conservation at the regulatory region of the genome,
That-
apart from the
Yeah.
protein.
Yeah.
So
Like in-
there will be
Yeah.
for the protein for the coding region of the genome, we would prefer using protein as a sequence, but for other regulatory regions and promoter
Like a
regions and others,
transcription factor binding site and
so
stuff like
for
that.
those we will because there is no protein there.
I don't know whether you do multiple sequence alignment. The question was multiple. I'm thinking that you can do blast with DNA at that point.
But if we just want to see if across, if
Across
the binding
several of them,
sites,
okay.
yeah, so it
That's
can
a
be,
chip there, right? Okay.
it can be useful.
Yeah. Good. Got it.
Or even without the intron-exon boundaries, a lot of mutations are there, I mean, accepted in the
No,
intron.
those things you juxtapose. That's why I was talking about
Right.
juxtaposition.
Yeah,
At
so-
the exon junction, you juxtapose. You cannot align.
we
But
mainly,
here you can align.
we mainly use it for protein level, but in certain cases we can use multiple sequence alignment.
There also, I think
for
you
DNA.
basically juxtapose because alignment DNA level में नहीं आता unless
No,
it's a
just
big alignment.
just
Yeah.
a regulatory region.
Yeah,
So
regulatory
something
region,
will come,
but yeah.
because it's very
So
difficult for
it
DNA
can do,
level.
yeah. DNA, that's the reason we're just debating this much because DNA is
It's a very complicated topic. So it's-
complicated topic. DNA varies a little bit. Alignment program, because there are only 4 bases, right? 4 bases. Even one goes up, the alignment won't be right. Okay. So I'll talk about the mistake that happened with the DNA alignment
Yeah, so
across
it's-
the protein. We'll talk about
it's
that
very
later.
difficult to align up DNA.
Yeah,
It's,
you
it's
have
very
to
difficult.
you have to know your signature very well. Once you know the signature, then you know the signature, like Tata box. Tata box, you know the Tata box. One, if you align it, Tata may not align with Tata. If you even take this much,
Yeah.
it'll never. You got to juxtapose them. So anyway, I think there may be other regions, significantly long regions. You could do alignment and find them. So anyway, so that's a good point you made. So anyway, that's why I'll stay away from DNA multiple sequence alignment. In DNA, you cannot use multiple sequence alignment for DNA,
It's
a
very
protein coding region. They'll be totally different. Okay.
Lot
One
of-
time, a student took the cDNA of all the proteins from the same family. Okay, same family and the multiple sequence alignment of the DNA. They were not even the DNA of the same protein because at the sequence level, they were coding for something else. It wasn't even same protein. So it was completely. So keep aware that when you're getting into DNA, it's a slimy slope. You can slip. You want to make sure that
it's a lot more variables come in than the proteins. So
Yeah,
it's
you
very
got to-
difficult to just map it.
Yeah, because just there is 4 and the mutation matrix is not that complicated.
So it will be like
yeah, so you know a
multi-dimensional.
little bit more about your signature.
It will become a multi-dimensional mutational matrix just
Yeah,
to code
yeah.
for that.
Yeah.
So it's really complicated there.
Yeah. So now you have, okay, so signature you are seeing. In signature, why are the signature not exactly identical? Because within the signature, still functionally tolerable amino acid went in there, right? So how do we know that? Like a serine to threonine is tolerated. We know that.
Similar nature.
How would the computer know? You're doing the alignment in the computer. The biggest problem for alignment, even the DNA thing you were talking about, is because computers can see and they don't know what they're looking for. We have to tell them what we're looking for.
Ma'am, when there are transitions and versus transversions, with
Yeah.
transversions we have like more deleterious effects. Sometimes they will code for like non-synonymous mutations are going to be there. In proteins also, like when we are using clusteral omega, they will use different color codes for positively charged, negatively charged, hydrophobic.
So yes,
like that.
so the
You want
full
to talk about transversion and transition?
conviction.
It's like, I think you're talking about purine and pyrimidine.
Yeah, that
Okay.
in the
Transition-
DNA.
transition
That's
is-
also
yeah.
slimy
But
side.
yeah, so
got
it's
it.
purine to pyrimidine transition is
Transversion.
transversion and
Purine to purine.
and purine to pyrimidine or pyrimidine to purine is
Transition.
transition.
Yeah.
So
I don't know. Yeah, it's a good question. I haven't thought much about it because I know they have different effect on the protein, of course. But when you're looking at the double stranded DNA, both strands can code. Here, transversion may screw up
The
because this is
The
a
mRNA.
purine and this is a pyrimidine. If you have a purine into pyrimidine, you lose the hydrogen bonding. So it has to be deleterious. That's the point you're trying to make.
Yes, ma'am, and their effect on the protein level as well. Sometimes it might not be in the third positions if you are following a particular ORF.
That's
Yeah.
Say in lysine to arginine, the function is pretty much conserved. But from lysine, if it goes to say threonine,
Yeah.
then it will be
That may not be transition necessary. It may be they just wobble into the middle and
Yeah,
the
so it
first one,
depends.
that
So
kind of thing.
we discussed it earlier. So most of the mutation happens at the wobble
third
base.
side. Yeah.
So even the mutation at the second base is deleterious.
Deleterious. Yes.
The purine
Yeah,
purine to pyrimidine mutation, we have sufficient number of DNA repair mechanism that will come because at the DNA level it's not pairing. When it comes to clean it will clean it.
So
It will delete it because it recognizes that there's something wrong there. It
Base
looks
pair
at the,
excites.
yeah, base pair is not right. It will come and delete it. All those with all those mechanisms and everything, we have accumulated so much mutation and so much function and everything. We still fall sick and everything, right? So I sometimes think of we should be really thinking how are we normal all the time. You know, sick is something, sick is rarity. We should all be sick and rarely we should be
but
normal.
our body is really smart. So we have a nature has preserved
Natural
some- yeah,
selection,
selection is
that's
there.
what it
We
is.
have a repair
And-
mechanism intact.
Yeah, so a lot of mechanism to keep us normal. Okay, lot of redundancy, lot of immunological system. Beyond all that, we fall sick. That's a different story. So now, because of the signature, now you guys must have already gotten a feel for why we are doing sequence alignment. That was the purpose of this lecture, this podcast, right? So protein can stay. Just to summarize,
Yeah.
are we done with it, or are you going to some
Yeah,
points?
almost because
Still,
then we'll have to discuss. So in the next podcast, probably we'll be talking more about multiple sequence alignment, the how we do homology,
Homology modeling, maybe.
yeah, with
Hmm.
multiple sequence
With
alignment.
multiple.
Just a brief introduction to that because it will come later on. We'll also discuss the other methods for phylogeny. So that will come up, and we'll slowly transition towards the HMM part. So that will be
Yeah. So the multiple sequence part we're going to do next. So what I'm going to just summarize what we did today. We learned what are sequences. There are two types of sequences, DNA and protein. RNA is another one, but that's not really different. So DNA and protein, two different languages. We know we can easily convert DNA into protein in a computer. So we can go a long step that nature does it so carefully, we can do it in the computer. Then from protein to function, we struggle. We struggle, and I think we made a point that we need sequence alignment to go from protein to function. Even there, we depend on known proteins. Known proteins, something is a kinase already known somewhere in yeast or any other system. If you know a kinase, it's a kinase. If it is a protease in some kind of a fly or something, it's a protease everywhere. So it doesn't have to be same organism. Someone has done that we are using to sequence element. So in a way, I feel like we are doing so much, and it's pathetic that we have to already know something to do something, So, but that's how it is. That's the state of the art now. But AlphaFold and everything is going to maybe change that because we're getting little smarter about protein fold prediction. So hopefully by the time you guys are my age or something, there will be no sequence alignment. Everybody is talking about structure. But today we got to bear with what sequences can do, because that also tells you a lot about biology. It's not just coming and predicting function. It's so much about biology, natural selection, what is tolerated, what's not tolerated. So anyway, with that, I'm going to end. Drupadi, want to say something? But this was good.
It was very nice.
We
got to
covered
discuss-
most.
We covered most.
yeah,
of
we
it.
covered most of what we wanted to do today. So
Okay.
Yeah.
Thank you, guys. Hope to see you guys again with multiple sequence alignment. Okay.
thank you.
Bye now.
Bye.
Bye.